really a leader in the area of mass spectrometry at the center to give the next talk. David?
Okay, thank you. Let me find my PowerPoint. Okay, do we see the...
You're good to go, David.
We're good to go? Okay, good, thank you. Okay, first of all, so there's something I need to adjust here. Okay, first of all, Stephen and Ned, thank you very much for inviting me to talk here today. Now, Chip and Lance have just given a really nice talk highlighting specific applications of proteomics in cancer. In this talk, I want to kind of change gears a little bit and give an overview of global mass spectrometry-based proteomics. And I'm not going to spend too much time talking about specific applications to cancer because this is not a technology that's, it's obviously not specifically for cancer, but can be applied to a wide area of research. So, for example, I am an associate director of the mass spectrometry facility at Rutgers, but my own research program focuses on the application of proteomics for inherited neurological diseases. So what I want to do in this talk is basically give a kind of a brief overview of some of the approaches and some of the methods that are being currently used in bottom-up quantitative mass spectrometry. So I don't need to really define global proteomics, but I will anyway. This large-scale characterization of the proteome from different cell lines or tissues or organisms. And sort of key aspects of this kind of global proteomics efforts is obviously the identification of proteins in a complex mixture, quantitation of the proteins, and finally, characterization of the proteins, particularly in terms of post-translational modifications, which is obviously a extremely important area of research in cancer. So my talk is going to focus on bottom-up proteomics, and there's also another type of proteomics called top-down proteomics, which is a method for the analysis of whole proteins by mass spectrometry. Bottom-up proteomics is a method for the analysis of proteins based on mass spectrometric analysis of proteolytic fragments of proteins. And bottom-up approaches are the most mature and widely used approach for protein identification and characterization. And that'll be the focus of this talk. So before getting too involved in some of the methods involved in global proteomics, I think it's worth pointing out that, and again, that was very clear from the previous talk, that proteomics and genomics are highly integrated in the analysis of problems in cancer. And a good example of this comes from the NIH Clinical Proteome Tumor Analysis Consortium. So the overall goal of this consortium is to identify proteins that are altered in cancer to provide information about potential pathological pathways and to help identify biomarkers that could be used to help diagnose disease and monitor response to treatment. So this consortium is taking two different approaches to identifying potential protein changes of interest. They have a targeted approach, where they're using information from a genomic data set, so information like mutations and genomic rearrangements, and using that data set to help identify proteins that might be of specific interest in cancer that can be addressed on sort of an individual level. They're also taking an untargeted approach where they are generating genomic data sets and proteomic data sets, and then analyzing them together to try and identify interesting candidates. And sort of the advantage of the latter approach is that this is a sort of more global approach and more likely to identify protein changes that might be missed from a purely genomic standpoint. But anyway, it's the integration of genomic and proteomic approaches is obviously incredibly important for this sort of effort. So now I want to speak a little bit more about the actual methods and the principles underlying global mass spectrometric efforts. So as I mentioned earlier, we focus mainly on bottom-up proteomics, which is an analysis of protease digested proteins. And this can provide quite a lot of information about proteomes, including obviously the identification and quantitation of individual proteins in a complex mixture. You can gain a lot of information about how proteins are processed, both in terms of proteolytic processing, different splice forms, and most importantly, these kind of approaches can provide a lot of information about post-translational modifications. And I think currently there's over 200 different PTMs that have been identified, and many of these, especially phosphorylation, are incredibly important in cancer. We can also measure steady state levels and turnover rates. And maybe the most important application is that it allows us to globally compare samples from disease like cancer and controls and identify changes that may be informative. So down to kind of the nitty-gritty of the actual mass spectrometry, let's start off by just sort of talking a little bit about how mass spectrometers actually work. So mass spectrometers analyze charged molecules, and samples are typically ionized within the ion source of the mass spectrometer by a high voltage field. Once the sample is charged, it can undergo separation, deflection, and manipulation by the mass spectrometer. Now, the mass analyzer is under a high vacuum, and it takes these ionized molecules and it separates them based on their charge to mass ratio. Then it outputs them to the detector, where they can be detected, not surprisingly, and converted to a digital output. The digital output provides structural information in terms of both the mass divided by the charge, the m/z, and it also provides some quantitative information in terms of the relative intensity of the different ions that are being detected. So bottom-up proteomics, we digest the proteins into individual peptides for the mass spectrometers to analyze. And the most commonly used protease for this kind of approach is trypsin. So trypsin is a very efficient protease with a high catalytic activity, and it generates peptides with a basic arginine or lysine at the C terminus. So although trypsin is extremely efficient, over half of all the generated peptides are less than 6 amino acids, and these are too small to be detected by mass spectrometry. As a result, other proteases are also used for bottom-up methods, and these have different specificity to trypsin, and they can be used either together with trypsin or in place of trypsin to increase the coverage of proteomes of interest. So what information could the mass spectrometer actually provide? So a protein sample is digested with trypsin to give the proteolytic fragments. And these can be injected into the mass spectrometer and ionized. And the mass spectrometer will measure the mass over charge, or M over Z, of all these different individual proteolytic fragments. So the mass over Z actually provides useful information in terms of trying to identify these peptides, but it's not enough to identify a peptide. So for example, here we have a peptide of 507.3 M over Z, and that mass can be assigned to a number of different peptides. So knowing the mass of the precursor is not in itself sufficient to actually identify that peptide. What we need to do instead is actually determine the amino acid sequence of each individual peptide. Then they can be assigned to individual proteins. So this is where we conduct what's called data-dependent acquisition. Now, in a typical data-dependent acquisition experiment, the mass spectrometer conducts the MS scan, and again, we see it identifies a number of different proteolytic fragments. And then it can actually isolate just one peptide out of the mixture that's going in at any given time. And it can fragment that precursor ion. So it takes this, it uses something called collision-induced dissociation by colliding the fragment with an inert gas like helium. And what this gives us is what's called an MS-MS or MS2 spectrum. So that the precursor has been taken and now it's been fragmented, and those MS2 fragments can provide important information about the amino acid sequence of the original peptide and can be used for database searching and assigning that peptide to the original protein. So how does the MS2 scan provide information about the amino acid sequence? Well, the fragmentation of the precursor ion, fragmentation of the precursor ion occurs typically at the peptide bonds, generating ions called B ions, which contain the N terminus of the peptide, or Y ions that contain the C terminus of the peptides. Now, the mass of these fragment ions taken together with the mass of the original precursor ion are typically sufficient to actually sequence the peptide, allow us to identify the peptide and map it to the original protein. So in a typical experiment, peptide ions are analyzed to obtain the precursor ion masses and the fragment masses via MS2, which provides the amino acid sequence. And then we use a search engine to match these MS2 spectra against theoretical spectra, which are generated from an in silico tryptic digest of a known proteome, and that known proteome is obtained typically by DNA or RNA sequencing. So that's how we can take the MS and MS2 data and match those to peptides, and in turn match those peptides to the original proteins. So if you take a complex proteome and conduct a theoretical tryptic digest on it, you will generate a lot of unique peptides. So for example, if you take the human proteome and do a theoretical tryptic digest on that, you'll get over 3 million unique tryptic peptides that could potentially be identified by mass spectrometry. When you take into account different splicing forms and you take into account different post-translational modifications, you'll have orders of magnitudes more unique peptides. And this is way more than any mass spectrometer can analyze in one go. So what we do instead is that the mixture of tryptic peptides is fractionated by reverse phase HPLC before it's injected into the mass spectrometer. So what this does is that it greatly simplifies the mixture of peptides being injected into the mass spectrometer at any given time. And it gives the mass spectrometer the chance to identify more of the peptides. So that's briefly how we do the protein identification. The other key component of global proteomics is actually being able to quantify all the proteins in that complex mixture. Now, there's a number of ways in which you can do that, but I want to just briefly discuss today 3 different methods that are commonly applied for clinical samples. I want to talk a little bit about chemical modification of the peptides to allow their quantitation. And I also want to talk about peak area integration and other methods that are label-free. So the first method I want to talk about is data-dependent acquisition with isobaric labeling. So isobaric labeling is a commonly used chemical labeling approach. And this slide here shows an example of something called iTrack4. So iTrack4 allows you to simultaneously analyze 4 different samples together. So if we have 4 samples here, which could be, for example, 2 samples from a control and 2 samples from a disease specimen, these are each digested with trypsin, and then the peptides are labeled with these iTrack reagents. So the thing about the iTrack reagents is that they're all the same mass, so that if you mix all these peptides together after labeling, they co-migrate on the reverse phase HPLC and elute as a single peak. But the reason they allow the peptides to be quantified is that they each contain what's called reporter ions. So we do a data-dependent experiment. Each are labeled with a different reporter. They're mixed together, and we conduct MS1 and again identify precursor ions. Then that precursor ion can be fragmented at the MS2 level, and each of the reporter ions can be measured. And the relative intensity of these reporter ions is proportional to the amount of this peptide that was derived from each of the individual samples. So this is how isobaric labeling works. It allows us to quantify peptides, and in turn, it allows us to quantify the proteins from which they were originally derived. So it's technically straightforward in terms of chromatography and mass spectrometry, and you can analyze up to 16 samples in a single run. You can pre-fractionate the samples after you've mixed them together to increase the proteome coverage. And you can actually analyze more than 16 samples if you do separate experiments using a bridging reference standard to allow you to compare those experiments at the end of the study. Disadvantages of isobaric labeling are it's expensive, and the labeling steps are additional microchemistry steps that add variability to the final data. A relatively low percentage of the detectable peptides are fragmented. And then a problem with data-dependent methods overall is that the selection of the precursors detected at MS1 for MS2 fragmentation is semi-stochastic. So that introduced some element of variability into the data, but it also means that if you do multiple experiments, in some experiments, you're going to see a peptide, and then just by random chance in the next experiment, you don't see the same peptide. So this gives us what we call missing data, and it makes analysis of multiple experiments quite challenging. And a specific issue with isobaric labeling is that there's a problem with quantitation called ratio compression, which is due to background reporter ion signal. So currently, the most widely used form of quantitative proteomics, especially for clinical efforts, is label-free methods that are based on quantifying the MS1 signal of peptides obtained from tryptic digestion. So what happens here is that we take our samples, we digest them with trypsin again, and we analyze them, and we do repeated MS1 measurements. So you remember the MS1 is the precursor ion, the whole peptide, and then we do this constantly, with occasionally measuring MS2 to provide the peptide sequence and allow us to identify those peptides. So once you've measured the MS1 profile of the peptides across the elution profile of the HPLC gradient, you could actually use that relative intensity to plot the elution of each peptide, and you can measure the area under each curve. And the area under each curve provides us with a relative measurement of the abundance of each peptide in each sample. So the label-free methods like this, you're using one mass spectrometry run per sample. So you're unlimited in terms of the number of samples that you can analyze. And clinical studies use hundreds or even thousands of individual samples using this label-free methods. It doesn't require the expensive chemical reagents for isobaric labeling, and it doesn't require the extra steps that introduce variability. It's accurate over a greater dynamic range than isobaric labeling, and as I've just mentioned, it's often used with analyzing clinical cohorts. And one nice thing about label-free mass spectrometry is that you can actually program the instrument to measure peptides from proteins of specific interest. This is an approach called targeted mass spectrometry. Disadvantages, because you're running one mass spectrometry run for each sample, label-free quantitation tends to be very instrument time intensive. You also need to have very, very stable chromatography for these kind of approaches. And finally, you still have the problems of missing data that I described for the isobaric labeling. So again, that's a feature of data dependent methods. So the last method I'd like to talk about for quantitation of mass spectrometry data is something called data-independent acquisition. So this is an emerging technology, and there's a lot of buzz about this method, and it potentially has a lot of promise for large-scale clinical projects. So in data-independent acquisition, we digest the proteins with trypsin. We fractionate them on the reverse phase HPLC and inject them into the mass spectrometer. And we generate the MS profiles. So again, each of these peaks here on the spectrum correspond to tryptic peptides. Now, in data-dependent methods, as I've described, we would isolate one of these precursors and fragment it with MS2 to identify it. With data-independent methods, what we do instead is that we isolate all of the peptides within a narrow mass over charge window. So in this window here, we've got about 4 or 5 different peptides, and they're again fragmented by collision with an inert gas. And then we measure the MS or MS2 spectrum. This time, instead of having one MS2 spectrum that's derived from a single precursor ion, now we have this whole mess of peaks, which is MS2 spectra derived from all of the individual precursors isolated in that M/Z window. So we can then use, there's ways to analyze this data to take this multiplexed MS2 spectra that's derived from multiple precursors and deconvolute the spectra and assign those individual deconvoluted spectra to peptides. And this multiplexed MS2 spectra also can provide quantitative data by integration of these individual MS2 peaks. So the next step is that the instrument then isolates another M over Z window and repeats the whole cycle. And it keeps taking different M over Z windows and doing the MS2 fragmentation until it's covered the whole range from, in this case, 300 to 1000 M over Z. And then it will start again from the beginning. So it keeps cycling with these different mass overcharge windows, performing MS2. So the big advantage of data-independent mass spectrometry is that you don't choose the MS1 peaks for fragmentation stochastically. Instead, you're going to take all of the precursors within that window and fragment them. So in theory, you're not losing any of the data now. So this may be a good alternative for label-free data-dependent acquisition for large clinical cohorts. And the proteome coverage using data-independent methods is also claimed to be greater than data-dependent methods. Technically, data-independent acquisition is quite challenging. You require very, very stable chromatography conditions. And as I've described, you have this multiplexed MS2 spectra that's derived from multiple precursor ions. It's computationally challenging extracting all the individual data from that multiplexed spectrum. So data-independent acquisition, as I said, there's a lot of interest in this method at the moment, and we'll probably be seeing a lot more of it in the future. So I think pretty much that's all I need to say about the methods and the principles underlying quantitative mass spectrometry. So I'd just like to finish with a few final thoughts. First of all, there's a number of different approaches to clinical proteomics, but key to the success of these methods is integration with genomic methods. The quantitative aspect of mass spectrometry is absolutely critical. Just worth mentioning that in a typical experiment, you can easily identify more than 10,000, identify and quantify more than 10,000 different proteins from a single cell type or tissue. It's also incredibly important that you're not only measuring and quantifying the individual proteins, but you're looking at both their isoforms and their post-translational modifications. And finally, it's just worth saying that these kind of global proteomic studies can identify changes which can provide mechanistic insights into disease, which can potentially identify drug targets, and which can potentially identify biomarkers for disease diagnosis, for monitoring disease progression, and also for determining the response to therapy. Okay, so on that note, I will finish here, say that I work over in the IQB, part of the time anyway, in the biological mass spectrometry facility. And if you have any more questions about mass spectrometry or interest in any projects, we'd be happy to talk to you. So I will end there.
Thank you, David, for a very, very insightful discussion, really tying together the potential of the mass spectrometry with genomics and what we heard in the first two talks. We have a few minutes for questions. People could raise their hand and someone will see them, or add something into the chat. I can't see the raising of the hands. Has someone raised their hand?
No, I think hands are firmly down here.
I've checked the participant list, gentlemen, and I don't see any hands up.
Okay. Thank you.
So David, as a practical matter, for people who want to come and use some of these methods on the ground floor of the Institute building, who should they ask for when they get to the door of the facility?
Excuse me. Probably either talk to Peter Labelle, who's the director of the mass spectrometry facility, or Haiyan Zhang, who actually runs the facility on a day-to-day basis. They would both be happy to talk to anyone about potential projects.
Yeah, and they've both presented in various IQB events in the past, and I know that they're very keen to solicit more business.
Yeah, and typically these sort of experiments we run on behalf of clients. There are some services where we allow users to actually get their hands on the mass spectrometers, but most of the time we conduct the studies on behalf of the clients.
So that's just the MALDI device where you have walk up, I'm assuming.
The MALDI, we have walk up. I think some outside investigators using one of the instruments for hydrogen deuterium exchange.
Great. Well, I hope this, thank you for a terrific presentation. And I hope this directs more business towards you. And I hope this event will catalyze the possibility of some collaboration between your team and the George Mason Group.
Yeah, and if nothing else, I hope people have got a sort of a little bit of a better idea of how, they hand over the samples and they get the data back. What are the steps going on in between?